AllerGen's Clinical Investigator Collaborative, or the CIC, offers a unique partnership with pharmaceutical and biotechnology companies. The CIC conducts phase 2 proof-of-mechanism clinical trials using potential drug candidates that are in clinical development for the treatment of asthma. The CIC is unique in its ability to conduct complex studies using human models of asthma and in a multi-institutional setting, also using proprietary standard operating procedures to ensure that the results are uniform across our 6 centers. In December 2016, CIC's allergic asthma researchers published a study evaluating the effectiveness of a new investigational drug manufactured by ONO Pharmaceuticals in reducing symptoms of asthma after exposure to inhaled allergens. The investigational drug, called ONO 6950, is a leukotriene receptor antagonist that blocks inflammatory substances called cysteinoleukotrienes. These leukotrienes cause narrowing and swelling of the airways and play a key role in driving the symptoms of asthma. ONO 6950 is unique in that it blocks cysteinoleukotrienes from binding to 2 receptor types. This is the first dual leukotriene receptor antagonist to be tested in humans. We conducted a multi-center, double-blind, randomized, placebo-controlled, 3-way crossover study testing ONO 6950 and comparing it to placebo, and also comparing it to an approved asthma medication currently on the market. The approved medication, Singulair, is a single leukotriene receptor inhibitor, meaning that it blocks leukotrienes from binding to only one type of receptor. The study was conducted in a group of subjects with mild allergic asthma who received treatment for 8 days and then were exposed to an allergen which was inhaled through a nebulizer. After allergen exposure, we measured lung function and the early and late phase asthmatic responses to examine constriction of the airways. We found that ONO 6950 provided the same degree of inhibition of bronchoconstriction as the approved single leukotriene inhibitor, Singulair. We also looked for the presence of inflammatory cells in the airways and compared to placebo. Only the dual leukotriene inhibitor showed significant anti-inflammatory activities in the airways. Although the mechanism of action of this new drug is well understood to inhibit cis-LT1 and cis-LT2 receptors, the biology of leukotrienes and the receptor expression in asthma is very complex. The results from this study are important because they suggest that there may be additional anti-inflammatory benefit in asthma when blocking 2 cysteinoleukotriene receptors.